Okay, folks, we have completed our background knowledge building of chemistry, biology, and biochemistry. So it's time to jump into this next section of material where we learn how drugs make their way from discovery to use in humans. Today, starting with preclinical drug development. So today we'll be learning about what happens to drugs before they reach humans. And so I'll be showing you this kind of timeline of drug development over the next 2 days of class. And today what we are focusing on is kind of this first chunk. All of the research that happens before the drug reaches a human. Oftentimes, this is called phase 0, in addition to preclinical drug development. We'll be talking about where it's done, who does it, and how is the work done. So where is preclinical research done? Largely, this research is done at a variety of colleges and universities. As you can see, based on the pictures that I have on this slide, folks, you'll notice that Detroit Mercy is shown. That is because there is scientific research being done right here on campus. I am doing preclinical drug development actually in my lab on this campus as we speak. So this is where much of the preclinical research is performed. Who is doing this work? Well, the largest part of the work is done by graduate students who are in graduate school trying to obtain a master's degree or a PhD, and postdoctoral students who've already obtained a PhD. A small portion of the work is performed by undergraduate students. They do a smaller portion of the work, of course, because undergraduate students are pretty busy taking a full course load. And also, undergraduate students are still learning how to learn. Most of the work is done by graduate students and postdoctoral scholars because they have more time to spend. A large part of the purpose of graduate school is performing research, right? Graduate students are also transitioning from learning how to learn to creation of knowledge. So it makes much more sense that they would be performing this research and creating this new knowledge. Finally, of course, postdoctoral scholars do a lot of this work. They are people already with a PhD, and they work in labs full time doing research, and they are more independent than graduate students, and they're often they play more of a leadership role within labs. Now, I figured I would do a little bit of a discussion about what graduate school is, because I don't assume that many of you are already familiar with this. I assume that you're familiar with how undergrad works, but not graduate school. So I'll talk about it just briefly so you have an idea. So what's happening in graduate school is you enter shortly after, or after a few years, or even after many years of obtaining your bachelor's degree. Now, there are kind of a few steps along the way. Now, in between your bachelor's degree and your qualifying exams, you take classes. And really, it's like half-time classes. You start to perform research. And you spend a lot of time learning about the subject you will be doing research on. And so this first stage of graduate school, a lot of learning is happening because you're still taking classes, you're just starting out in a research lab. Likely that research is new to you, so you're spending a lot of time just kind of getting up to speed on what you're actually doing. And then come qualifying exams. And this is when you officially become a PhD candidate. Qualifying exams, some people literally take exams, but oftentimes this kind of looks like a mini defense where you present your ideas to a couple of faculty. You say, this is the research I think I'm going to do, and they give you some ideas about, how it might work, how it might not work, for example. In between your qualifying exams and your thesis defense, there's usually just a lot of research. Most of the time you'll be done with your classes, and so the rest of your PhD, you're really no longer a student, you're working full-time in the lab. Sometimes you might be a TA for an undergraduate course or an undergraduate lab, but the vast majority of your time is spent in the lab, so working, 9 to 5, doing experiments and obtaining data. So I really like to think about this portion of your graduate work as, yet you're really no longer a student. You're transitioning towards being a creator of knowledge. And so once you've kind of built enough research and you have a story to tell, it's time to defend your thesis. So that's when you tell a story of what you've discovered to the world, really. And following your thesis defense, you really just wrap things up. Again, spelling is challenging. Let's try that again. You're kind of just wrapping things up until the next graduation date. You submit your thesis. Maybe there were some edits provided to you. Maybe you do a few last-minute experiments. But then you wrap things up, graduate with a PhD. And so that's what graduate school is. Finally, let's talk about how research is performed. So generally, the type of research we're talking about for preclinical drug development is scientific research based on the scientific method. And so you've likely learned about this in classes prior to now, maybe in high school or middle school. But really, the scientific method is research inspired by a question or hypothesis. So what generally happens is, you're curious about something. How do cells work? Right? Maybe that's your question. And then really what's going to happen is you do some reading. You educate yourself about how cells work. You try to find, what is not known yet and what is known. And so then you can turn your question into a hypothesis. And a hypothesis is really a question that is asked based on what is already known. It's a bit more of a refined question. Following formation of your hypothesis, you do experiments, right? You test that hypothesis. You design some experiments that you think will answer that question. Once you've done those experiments, you ask yourself, okay, what outcome did the experiment give me? You analyze it. You try to draw a conclusion based on your original hypothesis. Was my hypothesis supported? Was it not supported? Then you report your findings to the world and start all over again with another question. And so this is really, in reality, a very messy process. Research is not linear. It often branches off into all sorts of ways. And a lot of times when you do an experiment, it doesn't really work out the way you thought it would. So you'll start doing research by reading primary literature or peer-reviewed literature. From this literature, You form your hypothesis and make a research plan. Then what happens is you design some experiments, you perform them, you gather the data, you analyze that data. But a lot of times, experiments fail. Maybe your hypothesis wasn't asked quite the right way. So you do some more experiments to fix your hypothesis, or because your hypothesis didn't work out as planned, right? So more experiments. And you thought you were done, and you were doing all the experiments you thought you needed to, but no, there's actually more, because as you learn more through experimentation, you tell yourself, oh, actually, there's all this stuff that I still need to ask questions about. So you do more experiments. Finally, you write up your findings. Oh, look at all this amazing stuff that I found out. Look at all these experiments. And you submit them to a scientific journal for publication. But then, of course, reviewers come back and they're like, well, this is great work, except you didn't answer this question or this question. So you do a few more experiments before you can publish that work. So you do those experiments and resubmit your findings to a peer-reviewed journal. And that is the research process. Up next, we'll talk about what I mean by a peer-reviewed journal.